With this video short, I'm going to quickly recap the 4 levels of protein structure. Okay, so the 4 levels are called primary, secondary, tertiary, and quaternary structure. But what do each of those tell us about the protein? So primary structure tells you the linear sequence of the peptides in the polypeptide chain. These amino, kind of here depicted as beads on a string, which is also how histones and DNA look. But that's a side note. Okay, so it's the linear sequence. So A, M, K, T, S, Q, R, S, S, R, A, G, G, right? Just those one letter code amino acids. What holds those together, holds those residues, those individual residues together are amide bonds or peptide bonds. Peptide bonds is likely the more correct answer. Yes, they are amide functional groups, but since it's in a protein and proteins are called polypeptides, we call that bond a peptide bond. Okay. There's also limited rotation around the alpha carbons. And as you'll see in the notes, if you review the notes, those are called you have what are called phi and psi angles. And those limited interactions or rotations around the alpha carbon of each residue have to deal with steric hindrance of the side chains that are coming off, those R groups. Okay? You can't rotate freely so that 2 R groups are close to one another. That's going to be sterically hindering each other. So there are allowed angles at which the single linear chain could kind of turn and tweak and fold on itself. Okay? Which kind of brings you into the next level of structure, which is called the secondary structure. So secondary protein structure talks specifically about the interaction of the backbone atoms. So remember, in an amino acid, a single amino acid, the N terminus, the alpha carbon, and the C terminus are all part of the backbone. Those are the 3 kind of major atoms in the backbone: the nitrogen, the alpha carbon, and the carbonyl carbon. 3 big atoms in the backbone. The side chain that's coming off is the R group. Okay, so when we talk about secondary structure, that forms when you try to maximize the number of hydrogen bonds of the backbone atoms, not of the side chain atoms, of the backbone atoms. I cannot reiterate that enough. So secondary structure forms 3 common shapes. You see alpha helices, and they can either be right-handed or left-handed, depending upon which way they're turning. So if they're like turning with your right hand, with the thumb pointing to the direction the helix is going from pointing towards the C terminus, then it's a right-handed helix. It could be a left-handed helix if it's turning the opposite direction. So you see alpha helices. You see beta sheets, and in Pymol, the beta sheets are depicted by the arrows. And those beta sheets could either be one of 2 ways. They could be parallel or anti-parallel. Parallel, the arrows are all running together. And the hydrogen bonds of the backbone, right, because that's all secondary structure, is talking about the hydrogen bonding patterns of the backbone atoms. So if we're talking about parallel beta sheets, those hydrogen bonds are kind of kinked as compared to anti-parallel beta sheets where the hydrogen bonds are in a straight line. And way back in that first lecture, I asked, which hydrogen bond is stronger? Is it the one where the hydrogen's in the center in a straight line of the 2 atoms of the donor and acceptor? Or is it the one where the hydrogen's kind of kinked between the donor and acceptor? And the stronger hydrogen bond is going to always be in the straight line. So anti-parallel beta sheets are technically more stable, less energy, than parallel beta sheets. Then the third type of secondary structure you see is more unstructured. It can be turns and loops. So in Pymol, those are just the kind of spaghetti string coil. Well, not coil, but the spaghetti string pieces that connect alpha helices and the beta sheets. Okay. So again, secondary structure is solely looking at maximizing, when a protein's folding, maximizing the number of hydrogen bonds in the backbone atoms. What about the side chain? That's tertiary structure. So tertiary structure of a protein looks at the interactions of the side chains. So now we're talking about all the R groups. Remember, we've got many different types of R groups. Some are hydrophobic, some are positively charged, some are negatively charged at neutral pH, some are polar, some contain a sulfur, right? So how do all those side chains interact with one another? Okay. This can be through covalent or non-covalent bonds. There's only one type of covalent bond that side chains can have, and that is the cysteine forming a cysteine bond with itself, so a SS, covalently linked. And the cysteine bond is also called a disulfide bridge. That's the only covalent interaction you can see with the side chains. All of the other interactions that the side chains can have with each other are non-covalent. Hydrogen bonding, ion pair, also known as electrostatic. So that's a positive and a negative charge coming together close in space. Hydrophobic interactions, pi-pi stacking. That's where some of those amino acid side chains that have double bonds, so tryptophan, phenylalanine, and tyrosine, stack one on top of each other. So those pi electrons, or the electrons in the double bonds, are kind of almost kissing each other, essentially. What other types of non-covalent interactions can you have? We said hydrogen bonds. We said ion bonds. I mean, you could get really particular. You could say ion dipole. So maybe there's like an ion, so a positive charge, and it's close to the, like the carbonyl, the oxygen on the asparagine. So it's called a ion dipole charge. Again, back in chapter 1 lecture, I should have a slide that talks about some of those other not as commonly talked about interactions. But anyway, so tertiary structure, side chain interactions with each other. Anfinsen's fold, so, and then quaternary structure, we'll get to quaternary. So quaternary structure talks about the higher order oligomerization. Is it a monomer? Is it a dimer? Is it a trimer? Is it a tetramer? Is it a complex? Usually anything bigger than 4 things, we call it a complex. Is it a homodimer? Homo same, so is it the same subunit, dimer, 2 of them together? Is it a homotetramer? So 3 of the same things coming together. Is it a heterodimer? Hetero meaning different, dimer meaning 2. So is it like an alpha and a beta chain? So 2 different polypeptide chains that assemble together. As one of the slides showed in E. coli, only 20% of all proteins in E. coli exist in monomeric form. They work in a monomeric form. That means 80% of all proteins in E. coli have higher order structure so that you can describe their quaternary structure. All right, so proper protein folding is Anfinsen's experiment, which uses insulin. So he kind of denatured insulin with some chemical reagents, and then he allowed it to refold. And what he saw was that, or what he, I guess, came up with, the end takeaway line from that experiment was that the primary structure determines the tertiary structure. Okay. Remember, secondary structure is all about maximizing the number of hydrogen bonds of the backbone atoms. But if you have, let's say, 2 polypeptide chains of 300 amino acids, but they're in a different sequence, those amino acids. If proper protein folding only depended upon secondary structure, then those 2 totally different polypeptide strands with the same number of amino acids would have the exact same structure. And they don't. And they don't at all, because primary structure determines tertiary structure. So it matters. Not only does the number of amino acids in your chain matter, but the side chains present and how they can fold and maximize their interactions drives what protein folding, not to mention the collapse of that hydrophobic core. So in a protein that looks something like this, there's kind of a center, right? There's a solvent exposed area on the outside, and then there's a shell on the inside. And those hydrophobic residues, valine, isoleucine, phenylalanine, methionine, you can look back at that hydropathy scale. Those hydrophobic residues are going to want to stay in the core of that protein, way away from all this water out here around them. So the collapse of the hydrophobic core, which is kind of maximizing the number of hydrophobic interactions you can get, is what kind of helps drive that proper folding. Okay. So that's kind of a quick and dirty summary of the 4 levels of structure and some of those important highlights I wanted you to know.